So today we will discuss the structure of mitochondria. So what is mitochondria? Basically, mitochondria is a organelle. Mitochondria is a organelle which is present in our cell of every eukaryotic organism, right? So this organelle is known as ATP mill of cell. ATP mill of cell. So why it is known as ATP mill of cell? Actually, you know the process of respiration. That process of respiration occurs in mitochondria. So what happens in respiration basically? Let's discuss the overview, right? Respiration. What happens that glucose C6H12O6 plus oxygen, they form carbon dioxide, water, and ATP. And what is ATP? ATP is actually the energy currency of energy. Currency of energy. So this ATP is produced in mitochondria in the result of the respiration. So that's why this mitochondria is known as the ATP mill of the cell. Moreover, this mitochondria is the second largest organelle in our cell if we exclude the nucleus. Like after excluding the nucleus, it is the second largest. But if we talk generally, so number first is chloroplast. If we talk about plant cell, right? Chloroplast. Number second is nucleus. And number third is mitochondria. I said that if we exclude nucleus, then it is second largest. Otherwise, generally it is third largest, right? Mitochondria. Okay. Now let us discuss the structure of mitochondria, because these structures are quite important in order to study respiration. Like topics like topics are respiration, right? So let me draw the structure of mitochondria. So consider this is the outer membrane, right? This is the outer membrane, right? Here we are having finger-like projections known as cristae. Okay, here we are having, I'm not good at drawing, but these are something like this. These are known as cristae. What are these? These are actually the finger-like projections. Finger-like projection. Right? Now inside this we are having matrix. Let me fill this with matrix. Okay, this is known as matrix, right? And now here we are having, in this matrix we are having some ribosomes. Here we are having ribosomes of 70S, because 80S ribosomes are present in the cytoplasm, not in mitochondria, right? Then we are having over here circular DNA. We are having here circular DNA. Like this mitochondria contains its own DNA and ribosomes, right? Now next we are having here enzymes. We are having here enzymes for what? For what purpose, right? We are having here enzymes for Krebs cycle, right? We will discuss what are these. Okay, this contains another organelle which is known as oxysome. right? Okay, here is another organelle which is known as oxysomes. Now let's discuss what is now this oxysome basically. Let me draw the structure over here. This is a base palette, this is a stalk, and here we are having, right? These are known as oxysomes or F0, F1 particles, or not particles, only F0, F1. Now what does that mean? This one is known as F0, this is known as F1, and this is known as F0. These are named after the scientist who discovered them was Fernandez Morin. So it was discovered by them. Okay, what does this organelle contains, this particle contains? Basically it contains an enzyme known as ATP synthase. It contains enzyme known as ATP synthase, and ATP synthase is quite important. Why? Because it converts ADP into ATP. Like in the presence of this enzyme known as ATP synthase, this ADP is converted into ATP, and this process is quite important in the respiration. So this is the origin of oxysomes which are present in the mitochondria. Moreover, over here, like you can see, this whole space is known as intermembrane space. This is known as intermembrane space. This is known as intermembrane space, right? Now let's discuss some reactions over here. First of all, in respiration, like the first step is glycolysis, right? First step is glycolysis. Where it occurs? Basically it occurs in the cytoplasm, right? It occurs in cytoplasm. Second we are having is link reaction. Basically link reaction, what happens in it? Actually in the glycolysis, glucose is converted into pyruvate or pyruvic acid, right? So in this link reaction, pyruvate is converted into acetyl coenzyme. This pyruvate is converted into acetyl coenzyme, where actually it is converted into inner membrane in inner membrane space. This pyruvate is converted into acetyl coenzyme. Now third we are having is Krebs cycle. Third is Krebs cycle. It occurs in the matrix. Number fourth we are having is, which is the last step basically, which is electron transport chain. Right? So this happens in the cristae. This occurs in cristae, or you can say the inner membrane, not inner membrane space, but in inner membrane, right? So here you can see this respiration. So basically here is another enzyme which is present on the inner membrane. Here this is like in this cristae and inner membrane space. This is enzyme known as succinate dehydrogenase. This enzyme is known as succinate genase. Now what happens, like what is this? Let me write it here. Succinate dehydrogenase, right? So basically it is a enzyme which helps in the link reaction, like it converts this pyruvate into acetyl coenzyme, right? It converts pyruvate into acetyl coenzyme, but like this enzyme is helpful in this link reaction, right? So this mitochondria is it's controlled by semi-autonomous. It is controlled by semi-autonomous. Why? Like what does that mean? Basically, you can see over here that it contains its own DNA. It has its own DNA. It has its own ribosomes, but still it is controlled by nucleus, right? It has its own DNA, own ribosomes, own protein synthesis, own protein factory, but still it needs the signals from nucleus. So that's why it is known as semi-autonomous. It is controlled by a semi-autonomous system. So I hope everything is clear to you. If you're having any type of query, you can ask me. I'll be glad to reply you guys.